I'll just recap about registered reports. So registered reports are where we submit the pre-registration to a journal and get some feedback. The place where we left off, so we didn't have a lot of time to discuss some of the evidence, but I did want to show you that we're getting more and more evidence that registered reports are working as intended. So we have a lot of issues with publication bias and outcome bias and hindsight bias. And as you can see here, the problem is that when we look at the literature right now, all of the papers have all the predictions supported. Very rarely do we see any null effects. I encourage you to go, whoever your PI is, whatever your lab is, and look at their publications. What is the proportion between null effects and supported effects? Right now in the literature, it's something around 90-something %. And it's not going down. The only thing that's bringing it down is registered reports. So you can see here that when comparing 71 registered reports to 152, almost comparable in terms of all sorts of aspects, you can see that whether the first hypothesis was supported or not just like dropped by about half to less than 50%, which seems a little bit more reasonable. There are all sorts of reasons why I would expect this to go even further down. But this is already a lot better because it means that we're also publishing null results, and null results are important. So we talked about the example of a vaccination. You would want to know if you're running a trial whether the vaccination did not work. So just seeing when it worked 2 out of the 100 times doesn't help you because there's like 98 times where it didn't work. So you would want the null effects to also be there in the literature. So this is one example. Another example, evidence for registered reports is when they did a semi-randomized control trial where they blindly assigned the first part, so just the methods and the introduction without the results. Some of them came from registered reports, some of them came from normal reports, and then they asked evaluators to say, what do you think about the methods, the quality, the amount that we will learn, whether it's important, the creativity, and so forth. And you can see all the comparisons here between the different. So as you go higher, this is the difference between registered reports and non-registered reports. So as you can see, on a bunch of things, especially the methods rigor, it seems like registered reports do a lot better. And the reason for that, it's very simple. Peer review over the stage 1 allows reviewers to ask for a lot more things. So basically, it's not just the authors that are submitting things to the journal. It becomes this collaboration between the authors, the reviewer, the editor, all of them trying to get to the best plan. Many times, especially when we do a replication, they also invite the original authors to say, does this look like a good plan to you? Something we're overlooking? And then it sometimes becomes a little bit of an adversarial collaboration where they really want things to work. They want this to be faithful to what it is that they've done. They want this to be a successful replication. So we work out things together. So that creates better methods and rigor overall. If you want a good summary of the literature, this is by Chris Chambers, Past, Present, and Future of Registered Reports. And this is where he addresses a bunch of things about things that people worry about in registered reports. So he writes about misconceptions and the realities of registered reports. So whenever I ask people, why don't you do registered reports? Go to your PI, go to the professor you're working with and ask, why have you not changed everything that you do to register reports? I honestly do not understand. It's available, it's out there. Number one issue that I see is that the top journals don't do register reports. So we aim here at HKU, we really care about impact factor. And when we try to go to a high impact factor, they don't have register reports. So that might be one reason. But nowadays you have enough top journals like Nature Human Behavior or Journal of Personality and Social Psychology in my domain, social psychology. So there are more and more that are doing register reports. I was talking to a very frustrated professor last week in the neuroscience domain in our department, who is about to go for tenure very soon, very frustrated with not being able to get to like high impact factor journals and a lot of rejections and moving from one to the other. And you've got journals like Cortex, where Chris Chambers is an editor. So if I would be in neuroscience, I would just like register report, Cortex all the time. Good enough. HKU appreciates it. Everybody wins from that. So why are they not doing this? Why are we not moving there like all the time? If I would be a PhD student, this is the only thing that I would do. Guaranteed publications, lines on my CV with an in principle acceptance, because of a lot of these misconceptions. So now you've had a chance to see the registry reports. You know what these look like. So some people say, I cannot explore. If I do a registry report, that it means that I cannot do anything more than that. So the main counter argument to that, and it's kind of like a famous quote, is that pre-registration is a plan, not a prison. So you can explore as much as you want. You just need to be very, very clear about what it is that you pre-registered and what it is that you explored afterwards, only when you saw the data. So it doesn't limit you from doing anything. It just allows you or forces you to be transparent about which one was confirmatory and which one was exploratory. Some people say it would limit my creativity. I think if anything, it increases creativity. It allows some feedback from reviewers to look at additional things. It allows you to think about things in advance and not catch errors and all of that. Registry reports are only for single shot studies. So you just need to do like the one study and then you can't do like a multiple studies. And this is no longer the case, definitely with peer community and registry reports, which I'll talk about. You have programmatic registry reports. You can do this step by step. Each one of these steps will be a publication, but you can do this more than just like a single shot. And sometimes if you want, you can run multiple studies within one registry report. Some people worry about scooping. They say, if I have a pre-registration plan that's public, that's out there, somebody is going to steal stuff from me. I think, if anything, this protects you from scooping, because once you've done the pre-registration and it's public, you've got a DOI. This is in your name with a timestamp on it. So if anybody kind of like saw this and decided to run this because they have more resources and you can just say, I did this first. I was the one that introduced this to the world. So it makes it a lot easier to make such claims. If you just said, yeah, in my lab, it's he said, she said kind of thing. But here you have like real evidence. Can be gained by post-registering. This, I've never seen anything like this. This makes it very, very difficult. Registry reports are basically not supposed to counter fraud, but they make fraud a lot more easy to detect, because everything is public, everything is out there. With enough searches, you'll be able to find anything. Do you need a fixed plan? Not suitable for early career researchers. So I think Chris Chambers is the one that convinced me that you can do this with early career researchers. I've done this with an MPhil student, my first one. And he's the one, Chinyu, if you remember from his feedback from the second session, that said that he recommends this for early career researchers. All the people that are working with me are either students or early career researchers in their PhD. So seems like it really has benefited their careers and their ability to get positions and all of that. Registry reports will slow the pace of research. This is not the case. So all of the theses that you've seen were done within less than a year with in-principle acceptance. So if anything, this really made it a lot quicker to publication. And registry reports give reviewers too much power to decide what research is undertaken. This is not the case. There is some worry about editors making decisions, but this is not specific to registry reports. This is also specific when you already completed everything and then you send this to the editor and the editor rejects it. So at least here, when you have a plan, before you committed everything that you have to that plan, you can get some feedback from the reviewers whether this is suitable or you can make some adjustments. So I encourage you to go and read this article. Really well done, a good summary of the whole thing, and has been impactful in order to encourage people to do registry reports. Another benefit for this, I don't know how many of you have done this, but in all of the thesis aside from one that you went through, there was a peer community and registered reports public registration. So you could have gone there and saw all the peer review that we received and how we responded to this. So I think when you ask registered reports reviewers, how do you feel about reviewing for registered reports? They feel very, very engaged. So I took a bunch of screenshots from Twitter, share this here with you based on Emma's slides. So Lisa here, I've been asked many times to review the same fundamentally flawed papers as the authors shop around for a journal. I'd much rather put in the extra effort to review the registered reports before it gets to that stage. So as a reviewer, you can contribute, you can change, you can amend, you can fix all sorts of issues. Whereas if it's not a registered report, you can just the only thing you can do is really reject it or ask them to collect another set of data. And then it's like a whole new process. Great to give feedback that can be acted on. These days, I turn down most review requests because I'm fed up of making the same comments, but today I received an invitation to review a stage 1 registered reports, and I'm actually excited. To me, if you go on my website, and I encourage you to do the same, you'll see that there's a section about reviewing. I'll show you what that looks like. And in that section, I've committed since the middle of 2021 to mostly submit my papers to Diamond Open Access and community journals like Peer Community and Registered Reports. I feel it's an obligation for us to do that. If you want to hear more about why I don't like impact factors and why I aim everything for Diamond Open Access, then the HKU library has an open access week from the 23rd, and one of those talks I will give at the library. So you're welcome to come and listen to this if you want to. Here's my general approach to reviews. I always sign my reviews. I don't understand anonymity. I think reviews need to be accountable. I try to be a careful reviewer, so I don't just do things in 2-3 hours. I try and spend a couple of days being very careful and testing everything. I try to offer positive, constructive feedback. So I have not recommended a reject in 4 years, I think. There's no reason to reject. If I see something that's fundamentally flawed, what I do is I write, this is what I think needs to be done. If the authors follow this exactly, then for me, as a reviewer, I would grant you an in-principle acceptance. So I encourage also the editor to say, if they do 1, 2, 3, 4, then it's accepted. Believe in open science, I'm accepting of imperfect results and null findings and all of that. And then I have a very strong preference for registered reports. So I will prioritize. We become very, very busy. We have very limited time. I will mostly review registered reports unless there's a really good reason not to. I don't want to waste my time on reviewing the traditional things. More and more reviewers are doing similar things to what I'm doing here. So something for you to consider if you ever get to the point where you want to review, you want to participate. Reviewing for registered reports just feels a lot better. All sorts of other evidence. I think we'll just skip this. Barriers. Yeah. So we talked about the benefits of registered reports. Why would you consider to do peer community in registered reports? This is really a revolution. I don't understand why it took us this long to do this, but once it came out, when I saw this, even though it was new, it was kind of like a risk. I decided to shift almost everything that I do to this peer community in registered reports, and I'll tell you why. So Chris Chambers calls this Registered Reports 2.0 for many reasons. It addresses many of the weaknesses in registered reports. He does a lot of talks, and I encourage you to go and listen to some of these talks. He talks about this in a much more persuasive way than I do. What is peer community in registered reports? Peer community in registered reports is part of peer community in. This is what peer community in looks like. So peer community in is a supra journal that has many, many communities. It's by the community, for the community. It tries to go away from all the for-profit publishers in order to do everything that they're supposedly doing just with the power of community, so that at the end it wouldn't cost anything. They have a budget for the next 20 years or so. So within each domain, they do peer community in. So you've got peer community in ecology, in animal science, in neuro, in math, forest, wood, like whatever you want. And increasingly, there are more and more. The last one is organizational studies. I'm trying, together with a group from Society for Improvement of Psychological Science, to do one for psychology. So that's coming up. And basically what they do is peer review over preprints. So instead of doing the pandemic, for us to wait for 2-3 years of peer review in, I don't know, Nature Human Behavior to come out, what they say is first post this as a preprint, and then as the community will give you feedback based on that. Then finally, once this is endorsed by the community, so the community gives peer review, the recommenders say that this is okay, then you have the option of publishing in any of the 100 PCI-friendly journals. So I'll show you what that looks like. There's a whole thing that's happening here, and then you've got a list of PCI journals. So once you get the endorsement from the community, with no additional peer review, you can publish in any of these. So for example, in psychology, you've got advances in cognitive psychology. If it's animal science, animal. So a very long list of journals that have worked together with the community to say, if the community endorses this, then I will publish this with no additional peer review. So part of the peer community they started this peer community in Registered Reports. So all of the other preprints are completed preprints with data collected and analyzed and the discussion. Peer community in Registered Reports just implements Registered Reports within the peer community in framework. So by the community, for the community. And the reason why I think it's revolutionary is because it introduced a lot of things that we really need. I'll focus on 3 of those because they relate to you. So I really recommend that you, maybe after this course, after you see what registered reports are, sit together with your advisor and really think whether to structure your thesis this way, starting from MPhil to PhD. I think that this is the only way that you can guarantee that by the time that you're up for the job market, that you'll have enough publications. And I don't know if you need doctorates. Does the practical doctorate need publications or something? Nothing like that, right? It does? It needs a publication? Okay, yeah, so even better. So this is a way that you can guarantee a publication out of that. Does it say any journal? Does it need to be a specific journal? Any journal, even better. Okay, how to guarantee this sort of thing? So one of the things that I was really worried about when I worked with Chinyu is that it took 3 months to get the peer review back for the registered reports. And then if you have 2 rounds, it's like 6 months. And then by that time, the MPhil is over. So, or the funding is over, or you need to move to the next stage. What they have is a scheduled review. I think Tommy was talking about this a little bit from his experiences. So it's really a revolution. If you looked at the templates that we shared with you for the registered reports, there's a section in there that deals with a snapshot. I'll show you what the snapshot looks like. Let's just open this for a second. So all of you now have this shared for your specific target with your team. I'm just going to open the template that we duplicated for you. It's this one. Yes, so what you'll see here is that if we go down a little bit, it has this stage 1 snapshot. So basically here, it has what is supposed to be at the end only 1 page that has the research question or theory, has the hypotheses, has the study design and methods, has the key analyses, conclusion, and references. Everything needs to be very, very short. So it's 2 to 3 sentences tops. Doesn't need to go into a lot of detail. And what that does is that you submit this 6 weeks before you want the review to happen. And the way that I do this with my students is that for example, many of them need to graduate by, so we're talking about undergraduates, they need to graduate by middle of April. They need to finish everything and have a grade by middle of April. In order to do this, we aim for peer review in the middle of January, end of January. So what we do is just before the end of November, we submit this snapshot. They go and they recruit reviewers. They show them the snapshot and say, this is the kind of paper that we need reviewers for. And then they say, we need you at the middle or end of January to be free for 1 week in order to review that. So everybody aims for that middle end of January. And then they have 1 week in order to do the review. So instead of just people sitting on papers and then taking a very long time and peer review taking for some strange reasons 2 or 3 months, that basically nothing happens because the actual review is 2-3 hours. Everybody just schedules this according to the snapshot. And while they're recruiting the reviewers, I work with my students within these 2 months, 6 weeks, in order to complete everything that we need in order to accomplish the stage 1 manuscript. So they review this for 1 week. Then the editor, the recommender, does this for 1 week, and then we get peer review within 2 weeks. What's also exceptional about peer community and registered reports, for the most part, is that they give you a decision letter that has actionable items. So it's like, do A, B, C, D. And if you do that, it will be accepted, in principle accepted for stage 1 for you to proceed. So within a very, very short time, so by the end of middle of March, end of March, we can already get an in-principle acceptance. Then I collect data very fast. I do this within 1 or 2 days online. And then I give this to my students. They have another week to analyze the data, another 2 or 3 days to write the discussion. And then the whole project is over and the thesis is finished. So all of the theses that you've seen were completed in this way. Scheduled reports, scheduled snapshot, peer community and registered reports, scheduled for somewhere in January, February, sent to peer review. We get back everything. We do another round. We send this back. End of March, we already have the in-principle acceptance, and then we proceed to do the data analysis. Masters, here it's a little bit more, so they have until the end of June, so we do this a little bit differently. But what this means is that you can, within 6 months, get your paper not only in principle accepted, but also completed all the way through. So Timothy is going to talk about in his talk, the upcoming talk, he's going to talk about how he completed the whole thing within less than a year. So one advantage is this scheduled review within 2 weeks. It's really a revolution. I don't know anybody else that does this. Only for that, it's worth aiming for this. You don't need to commit to a journal, so I'll just show you the list of journals that you can aim for. Yeah, so if we go to peer community and registered reports, which journals are supported? You have 2 types of journals. One of them is, so you have 2 categories, 2 categories. One is called PCIRR friendly journal, and these are journals that will accept your stage 2 without any additional peer review automatically. They don't even need to look at the plan or anything like that. Just by the community saying that they'll accept this is already accepted here. I think my favorite so far from these is Collabra. So most of our publications from peer community went to Collabra. They give waivers. So it's an open access, but it costs money. It's not Diamond. So you would need to pay money for APCs, but because you're early career researchers, they have a commitment that you can ask and get a waiver in advance. So you don't have to worry. You can ask for this, and they will probably grant this to you because you're a first author early career. So Collabra is a really good option. If you're doing anything that's meta science or methods related, then advances in methods and practices in psychological science is a very high impact one that is worth aiming for. If you're doing neuroscience, then Cortex supports this. So if I would be you, if you're in neuro, then as a student, I would just do peer community, Cortex. This is the best strategy for your career that I can think of. There's also other things. A lot of people quit Elsevier journals and form their own neuroimage reports. I don't know if you've heard about this. thing, they were upset about Elsevier starting to charge $3,000. So they just, the entire editorial board just quit and started a new open access journal. So if you're in neuroscience, neuroimage is a good option. So you've got like a bunch of other things, Royal Society of Open Science. All these are like automatically accepted. So you can consider those, whatever your field is. And then they have all the restrictions about if you need to pay APCs, if they have other things, if they require something, I don't know, a certain length, like 5,000. So you can just like go through this list and see what's possible over there. So this is already a revolution. So you just do this once with the community, you know exactly, you can ask for the recommenders, you can suggest the reviewers, you can do a snapshot with scheduled report, and then you can select any of these journals. So this is category number one. Many of these journals, aside from Cortex and advances in methods and practices, some of them are kind of like on the lower part of the impact factors, which is something that HQ cares about, which is why they also have the Registered Reports interested journals. And over here, you have some familiar names. So for example, Nature Human Behavior or Journal of Experimental Psychology Journal. So these are on the higher end of impact factors. And what this category says is that they don't accept this automatically. They get notifications from peer community. And when they see something that's relevant for them, then they reach out to the authors. So happened to us a few times that suddenly some editors contact us and say, we saw your replication, we saw your registered report, would you consider publishing this with us? So if you aim for any one of those, you can even contact them in advance and say, this is the project that I have. I want to do this with peer community registered report. Is this interesting for you? Or if you have a stage one, you can just submit this to them and then they'll tell you if they're interested in this or not, you know, before the data collection. So that's another option. Hopefully this will grow. I've been working with some journals in order to get them to join to peer community. So I know that some journals are upcoming, like International Review of Social Psychology, if you care about this region, then Pacific Rim, and some other journals from Asia Pacific. Yeah, so you don't need to commit to a journal. So you can just go to the community. And then next year, when you get the stage 2 acceptance, you can decide if you want to go to Cortex or Neuroimage or Collaborative Psychology or any of the other journals there. It's completely up to you. And then another thing that I really enjoy, because I used to get some very hostile reviews on the replication, original authors sometimes get very nasty, especially if they can hide behind anonymity of peer review. Here, as the author, you can actually select for the reviewers to be identifiable. The benefit for the reviewers, so if you are a reviewer, you say, why would I want to be identified? Is because all of the peer review is citable. So if you've reviewed for them, you can add on your CV, I've reviewed for this manuscript and here's the DOI. So you can get credit for that, not just something anonymous and then you write something on your CV and nobody can verify this. Here, everything is verified. So both for you as a reviewer and for you as an author, this is a big change that I strongly recommend. This is the magic of the snapshot. I think we talked enough about this. So it just like really narrows down the time that it takes to get to an acceptance in a registered report. All right, there's more benefits, levels of control. I'm not going to go into this. I'll just mention that yesterday I submitted a request from Teaching Development Grant to run this for an additional 2 years, but I still have some money for that. So we have something called the Hong Kong University Registered Report Challenge. Basically, what this offers is that if you submitted peer community and registered reports and got a stage 1 in principle acceptance, then I will be able to conduct, if it's like a Qualtrics, I will be able to conduct data collection for you up to 8,000 Hong Kong dollars. You can talk to your PI and say, I need 8,000 Hong Kong dollars, and your PI is like, I don't have 8,000 Hong Kong dollars for you. You have to do what it is that I want to do, but you have your own project and you want to pursue your own directions, no problem. Get a stage 1 in principle acceptance from peer community and registered reports. And if I still have the budget, especially if I'll be able to renew this for 2 more years, then I can do the data collection for you up to 8,000 Hong Kong dollars. I don't know if you have a good understanding of what this kind of budget can be used for, but I'll just tell you that with my students, almost all of the theses that you've seen are experiments between 5 and 10 minutes on Amazon Mechanical Turk with participant pool we run 600, sometimes 1,200. So depending on the length, we can go between 600 and 1,200. So for you, this is definitely a very high quality, well-powered paper. So as long as it's decent enough, like not half an hour, but something that can be run within 10 minutes and can be run online on Amazon Mechanical Turk with Qualtrics, then I can run this for you. So if you want, send me what it is that you're planning. There's a whole procedure. I can tell you exactly how this works. But this should encourage you, or at least your the professor that you're working with, to give you funding to try registered reports so that you don't have to rely on your advisor. Okay, so just know that it's out there if you want to talk to me about this. We still have, I think, 5, 6 openings for this. And if we'll get the second round, we'll have 30 more. So come take advantage of this if you want. So it's not like hypothetical. This is real, like we've done this. So what did we do? What did I do with my students? So we've got like 4 of these published already. So I've shifted everything in the last 2 years to these directions. All of the other thesis students, so we have like 19 of these in addition, so 23 overall. So you can go and check any of their, just like you did with your RR assessment, to check any of their projects to see what it is that they've done. And then we've got 3 more that are ongoing with an invitation to revise and resubmit that hopefully by the end of the year, we'll be able to wrap these up. This is a summary. So the person here, Yikang Zhang, who is in Maastricht University, joined the students in my courses. So these are the 4 students and the teaching assistant afterwards. So he joined in as the early career researcher at the beginning to submit the stage 1 register report to the journal, and then finally within a few months got this published in Collaborative Psychology, which has been very good to us, this journal. So this is what he writes. When I asked him for all kinds of feedback, he said, this is a very fruitful experience. I have learned quite some techniques to improve the transparency of my research. When you do this with peer community, you're not alone because the community is with you. You get some really good peer review. And then, running registered reports as an early career researcher would not be only feasible, but also highly rewarding. So learned a lot, pro open science, support and feedback, and feasible and highly rewarding is my summary or his summary. I can give you some examples. You've done our assessment, so you know what one of these things look like. But I thought maybe I'll give you just like the gist of it. So if you want, you can ask me some questions about different aspects. I don't know how far you went into the OSF and what it is that we shared, but I'll show you some example. This is the latest one. I have a bunch of stories to tell about this and everything that's happened since it was published. Yeah, so this is a classic study by Soman. And what this paper is actually about is that it talks about sunk cost effect. So I think most of us are familiar with this sunk cost effect. It seems like people don't only look at the future when they make decisions about net benefit, but they also look at what they've invested in the past. So if you've already committed to a certain course of action, then it's much more difficult for you to change that course of action. You kind of limit your recognition. It seems like something you're investing in. You don't see any alternatives, and you just want to keep investing, even though it's very clear that this might not go well. And we give a lot of examples. The person who I worked with during my PhD invested his entire career into sunk cost effects. So maybe there's some sunk cost effect over researchers who do sunk cost effects. And he studied this in all sorts of contexts, has shown this, so starting from projects to career choice, to relationships. You can see this in many aspects. Now, what someone came and said is that we have sunk cost effects for money, but not so much for time. So he demonstrated this in all sorts of different ways. I'll show you the stage 1 and stage 2 in principle acceptance. This is what in principle acceptance look like. This is the in principle acceptance for stage 1. And this is the official stage 2 endorsement with the results. So in this case, we were very lucky. I think about half of the peer community and registered reports that we've had were with Chris Chambers. So he definitely knows how to do registered reports, and here also very supportive. What you can see here is that it gives, so this is the recommendation before the data collection. So he wrote a summary of what it is that we were trying to do here. So in the current study, we propose a replication of 3 studies from someone asking whether some costs are weaker for time than for money. And then it tells us, if you complete this, these journals will automatically publish this for you. Because we don't have any data yet, it's level 6. So this is the highest level of control. So highly confirmatory, nobody has looked at the data, nothing exists. Another interesting, curious thing, and I've never seen any journal do this, is that he gave a URL to a pre-registered stage 1 report. So basically they did this for us. So once they've accepted, in principle accepted things, they registered automatically in a frozen timestamp that nobody can ever change. With our names, with the contributors here as the names, with a confirmation that peer community gave us an in-principle acceptance that everybody can see and is public, and it links to our manuscript and is associated. So if we go back to this, I'll tell you about the withdrawn here, part of the many stories that I have about this project. And you can see here that this was actually done by them, and this was done by us. So once they've done this, a little bit later that day, 3 hours later, I also froze the entire OSF page. So basically what I did was go to registrations and then do an open-ended pre-registration. So even if you don't do this, peer community and registered reports will do this automatically for you. Many journals, I don't know why, but they don't do this. They rely on you to do the pre-registration. And this is done automatically. So that's one nice thing about this. So the journals that you can publish in. And then what's interesting about this is that you can see the decision letter. Here's Dilip Soman that gave us some feedback as the author. So we'll just open this for a second. So you can see what the original author says. He also only had, so this is this is the second round. So he says, the only suggestion I have is this, but I look forward to seeing the projects move forward. You can also see, somebody very well known in the judgment and decision-making area. But then we had other reviewers, and you can see the round 1 as well, where we had 3 reviewers. Not all of them came, 4 reviewers. Yeah, so we've had a lot of review. You can see not only their review, but you can also see how we responded to these reviews. So if you click on this, yeah. And one of the things that I do, which I really recommend for you to do as well, is that whenever I submit a revision, I allow the reviewers to see everything that I've changed from the previous submission. But sometimes it's really difficult to keep track of what's been done. So I try to do this as structured as possible. So if you want, one of the many templates that we have, where is this? Yeah, so in these resources on my main page, on these resources, we have a template for how to respond to a decision letter. So basically all the decision letters that we have in our projects follow this template. And basically what's on this template is that we have a summary of changes table, and then there's some instructions to help people what to do and what not to do, and then copy paste where you have the note that was given by the editor or by the reviewer, and then you've got the response. Sometimes we split this to a response and the action. So for example, this is how we explain what it is that you've asked, and the action that we took is this. So I don't want the reviewers or the editor to look for any information. I want everything to be available and open for them so that they can very easily see that we've implemented everything that they said. So one other thing that I do is that I include the track changes, and then I do things on draftable. So the way that draftable looks is this. It basically opens the 2 next to each other, and then it highlights all the things that were changed, so you can see exactly what's been done. You can also do this with track changes in Word, but they don't like open this very, very nicely, and they don't align. So if, for example, something changing so you see over here, the alignment of the pages changed, but if I scroll this down, it kind of like continues from where it was before, and then it picks up. So it just makes it very, very easy for the reviewers and the editors to do this. So something I recommend for you to do. And then you can see for every comment that we make, we just like write what our response was. So that's another great thing that you can see in the stage 1 in principle acceptance. And then finally, this is what the stage 2 looks like. In the first round, in the first submission, we actually also added comparison between the stage 2 that we submitted to the accepted stage 1, so they can follow how we change from the simulated data to the real data. So just makes everything very transparent and clear. What I wanted to show you was the stage 1. I think all of you saw like a completed thesis and manuscript, but I wanted to show you what a stage 1 looks like. So I'll open this one instead. So what I wanted to show you here was the results section. So this is a stage 1. So all of this is random simulated data. And if we go down, you can see results. To analyze the effects, we ran this ANOVA, we conducted a 2x2, and then all of this is simulated data, so it doesn't show any effects. So at the end, the only thing that we needed to do is get the completed data, the collected data, plug it in the R markdown, and then run this again, and then just change the results section and add the results on top of this. Another thing that I wanted to show you, if you're not proficient with R Markdown, then I think it's kind of interesting to see how we did this, if you haven't looked at this already. So all that we do is with this R Markdown that actually becomes this HTML file that is run on the simulated data set. And then the second stage is basically the same kind of R Markdown. It's this one here. Data analysis, we only changed it to look at the raw data sets instead of the simulated data sets. What does an R Markdown look like? I'll just download this so you can have a look and see. So it looks a little bit like this. It has all these headings over here on the left, but it runs for every possible thing that we aim to do after the data collection. It has the code, so you can see the code and you can see the output. So it just makes it very, very clear with all the plots, how we're going to create them, what's going to happen from the beginning to the end. So it takes away all the guesswork. Everybody knows exactly what needs to be done. So I thought maybe I'll just show you on JAMOVI and JASP how to look at these from the different perspectives. So we talked about this very briefly. What are the different approaches that we have for null hypothesis? So basically what it is that they found, right? In the target article, what it is that they found is that they found no differences or very, very small differences between the 2,000, 20,000, 200,000, right? Let's say that this is our prediction. How can we test this kind of prediction? If we don't find support, we tested your model, we don't find support for any differences, what do? Can we conclude support with a null hypothesis? We cannot. If we use null hypothesis significance testing, there's no way for us to quantify support for the null, right? So what are two possible solutions to this? How to approach this? So we talked about Bayesian analyses, right? And we talked about equivalence technique. So in R, it's a little bit more complicated, but I wanted to show you first, if you've never seen JAMOVI or JASP, this is your opportunity. I'll walk through this very briefly. JASP, the reason why I'm showing you JASP as well, because JASP specializes, was built especially in order to get people addicted to Bayesian analyses. It's this team in the University of Amsterdam that's doing a really good job. JAMOVI is good in some aspects. I use JAMOVI a lot because it has automatic export to R, which is very, very easy for me to do things quickly. But JASP is becoming a lot more beautiful and powerful, and I'll show you a little bit of this. So if you go here and you do open and you go to data library, it already has a bunch of data sets. Because we're mostly focused on t-tests and ANOVAs in our design, I'll do this here. But of course, it has things for correlations and repeated measures and CFAs and whatever it is that you feel like you want to do. There's like stuff for that, but we'll focus on tooth growth, which is the data set built in R in order to demonstrate an ANOVA. I'll just tell you a little bit about what this data set looks like. So if you double click on the variable, then it opens up this thing and it shows you what the measure is. So it has continuous, ordinal, nominal, and then whether it's decimal or integral, then it has missing values and levels. So what you can see here is that this is length. So this is our DV. This is a continuous measure. Then we have SUP. And SUP is 2 levels, 2 categories, 2 conditions. And then we have dosage, 500, 1,000, 2,000. So we have 2 IVs. One of them is 2 categories and one of them is 3 categories. And then we've got a continuous variable. So very similar to most of your designs, right? So in your designs, what you have is 3, some of you have 4 conditions. Some of you try to do a 2 by 3, like you added another extension or something like that. So this basically takes care of all the designs that I've seen that you've done more or less. What is the first thing that we do when we have a data set? We do descriptives and we plot a little bit in order to see what the variables look like, right? So we can go to exploration and we can press on descriptive. And what you see is that immediately it gives you this APA style friendly table that you can just copy paste into your manuscripts. So just by doing 3 clicks, you already have all the descriptives. So this is something that you can already take, copy into, so if I, for example, open Word, then very easily I can copy this or export the table and then go here and just like paste this and everything is like ready. And then I just have to play around with what it looks like. This is not how it works with SPSS, so I don't know what it is that you're used to, but this just like makes it very easy. If you want to change what statistics are included, a bunch of other clicks, maybe I don't need minimum or maximum, maybe I need standard error. And then one of the nice things is that you can really do like a violin plots that are quite nice. So within a few clicks just gives you all of this out of the box, right? So within 2 seconds, I was able to see the data if I want. I can also do like a histogram and add a bunch of other things. So we could do this way, that way. I can just, see all the data and make sure that everything is decent and that I understand it and I know exactly what's happening. If there's like issues of normality that I need to consider, is it skewed or whatever else is in there. So this is the exploration. Now, if we want to run t-tests, so in one of the conditions, in one of the IVs, independent variables, we have 2 conditions. So if we have 2 conditions, we can run a t-test. Because these conditions are either this or that, it means that this is an independent samples t-test. So we run independent sample t-test. Once again, super easy. I just put this here, do this up here, and I've got the APA table here. But sometimes I want a little bit more, so I do mean, effect size, confidence intervals, and already I have everything that I need in order to proceed. Generally, now we try and move from student t-test to Welsh t-test because it's more robust, but there's not that much of a difference between them. You can see that the difference is like, where is this? Yeah, here. The statistic is pretty much the same. The p-value is about the same, but sometimes there are differences depending on all sorts of characteristics. But in a very quick, a few punches here and there, I was able to do an independent sample t-test. If I wanted to do a Bayesian, independent sample t-test. So sometimes it could be that I want to see whether there is support for the null or support for the alternative. I can do the exact same thing. So it's the same interface. I put this here. And then the only big difference, I guess, is that you need to kind of like define a prior. This is something that Bayesian has. So the whole idea of Bayesian is that you have some kind of preconceived notions about whether you will find an effect. Is there going to be an effect? Not going to be an effect? And then we call this a prior. And the posterior is that if you take the prior, so these are your prior beliefs, and then you take the data that's in front of you, what is going to be your updated prior, so the posterior after that. And then you've got like this really nice graphs, and you have two numbers. One of them is this BF10 and the BF01. BF10 basically says support for the alternative, and BF01 says support for the null hypothesis. What is this one thing? If there's one, if it's equal to one, it just means that the null is as probable as the alternative. So we don't have a preference for the null or the alternative. They're as likely. So we don't know. When we get one, we don't know. But if we get something that starts to deviate from one, the higher it is with the BF10, so the higher it is, the more likely that the alternative is from the null. So this is a little bit like null hypothesis significance testing and p-values. But if it goes under one, the closer that it gets to zero, the more likely the null hypothesis is. So I'll show you, I know it might be a little bit fast and confusing. I'll show you this with some data. Now it really doesn't show very much. I'll show you in a bit what that looks like. So I just wanted to show you that there is a Bayesian t-test, and it looks exactly like the independent t-test. So if we go up, this is the independent samples t-test, and it's kind of like the same interface. If you look at these two interfaces, it's almost the same. The only thing that is different between it is the prior. And sometimes, there's a default prior that many people use and are able to justify that. I won't go into detail what this prior is. For now, just accept it as a prior of what is the likelihood for the null or for the alternative. This is something that's kind of balanced on average. So if you don't know what your prior should be, you just use the default here. As you get more advanced with Bayesian, you might also want to change the prior. But this gives you the ability to define what your prior is and make it a little bit different if you want to. Now, how do we do the ANOVA? So I'll go here and I'll do the ANOVA. And what you see here is that we once again have the dependent variable, and then we have fixed factors. So already you can see a nice model. What do we see here? That in terms of null hypothesis significance testing, so what you can see here is that we have all the things that we expect from an ANOVA. So we've got the degrees of freedom, and we've got the F statistics, and then we've got the p-value. But we want to add a lot more to this. So typically we need a eta square or partial eta square. So we'll add this. We have a model, so everything is included in the model. Sometimes we want contrast or post hoc test. So post hoc test, basically, if we include all of this here, what it does is, maybe a little bit smaller. Okay, let's just keep it like this. What you'll see here is that for each one of the IVs, so we have an IV that's suck and we have an IV that's dose, it shows you what's called the main effect. If we look at this main effect here for suck, it shows you that the difference between OJ and VC is this, and what is the P with the adjustments from 2P. And then we have this for dose, so it compares each one of these conditions to see what the difference is. And we can also add a coherence D and confidence intervals that takes all of that into account. And finally, we also get this for the interaction. So you can, for each condition within the 2x3, for every one of the 6 conditions, to compare this to all the other conditions. So if you want to know if on the same level of SAP, OJ, what is the difference between those 500 and those 1,000, then you can go and have a look at this here, and then you can see the p-values with an adjustment, 2p adjustment. If you want to shift this to some other adjustment, or don't have any adjustment, then you can do this, or you can include both of them, and then everything is just ready for you in this. Another nice thing is that you can very easily plot to this. So you can add both of these here, and then it creates this very nice and easy plot. And then you can add a nice table and observed scores. So it will have also the jitter plot. So already when you've done this, this is already ready. It's like you can copy paste this and put this into your manuscript. So when I work with undergraduates and they don't have very sophisticated R skills, then basically they take this and they put this in their manuscript. Sometimes, we add the comments. So this is the comment. Then I ask them like, what IV, what PV, what hypothesis, and so forth. So you can add a bunch of things in here. Why is this better than SPSS? Because it saves everything together in one reproducible file. So I'll show you what that looks like. So on the one side, you've got the data set, and the data set is here. And then you've got all the analyses here on the left, how they were executed. And then you've got the results here. And then you can save the whole thing with one file. I'll save this in order to show you. So if we just go to like downloads, tooth growth, and I save this, and then I go to like downloads, then you'll see that we have tooth growth .onv. Now, the nice thing about this is that this .onv is actually a zip file, so you can extract this. I don't know if you're familiar with JAMOVI, just like doing this. So if we go into the file, you'll see it actually has all the analyses. So even if somebody that you work with does not have JAMOVI and they want to see your analysis, they can do this because it's just an archive. So you extract the archive, you press on the index, and then it just like has all the other things. So even if sometime in the future, like 20 years from now, somebody wants to look at your analysis, but it doesn't have JAMOVI, it's end of life or something like that, they still have the HTML file that hopefully will last a little bit longer, but it's just executable on most operating systems. And then, of course, you've got like all the images. So if you want the images, everything is just like there and reproducible. So everybody can find this. It's not exactly the case with SPSS, SPV, or whatever it is that it sends. Okay, so we've seen an ANOVA here. Move stuff. The one thing that I wanted to show you is just like you have ANOVA here, you can also have Bayesian ANOVA. If you don't have this, or if you don't have this, you can see it's called Bayesian JSQ. You go to modules, you go to library, and then somewhere here, there is, Bayesian methods, JSQ, you install it, and once you install it, you get like the stuff on the top. That also includes the Bayesian. So if you want to run exactly the same thing with a Bayesian ANOVA, you do this exactly the same. Both of them are fixed factors. You want to see the effects, you want to see the descriptive, you want post hoc. So I'm going to do the same with these two. I also want some descriptive plots, and they do really nice plots with this one. Credible, interval. And what you'll see here, and this is a little bit different from the other one, so it requires an understanding, is that you can compare this to the null model, or you can compare this to the best fitting model. So in the comparison to the null model, the null model is 1, and then you can see how likely everything else is compared to the null. So if we look at the interaction, so this is sub and dose and sub times dose. So this is basically the interaction. How much more likely is it from the null model? If we didn't have an effect, what we can do is we can compare this to the null model, and then we'll see that the null model is a few times more. So we can shift this to this one here, and then we'll see, so you can see compared. So this is support for the null model. You can see that this is basically 0. So you can just like get all this. Just remember that one is null and alternative are just as likely. If you use BF10, then if you go down to 0, it's more likely to be the null. If you go very, very high, it's very likely to be the alternative. I went over this very, very fast. I know it's not that easy to maybe understand all this, but if you play around with this, you'll understand a little bit more, and there's some really good tutorials. do you understand the main concept? Do you have any questions about Bayesian? So basically, instead of just testing the rejection of the null hypothesis, we compare the null model to some effect, main effect or an interaction. And then we can see, not just quantifying whether we can reject the null hypothesis, but we can quantify also support for the null hypothesis. Okay, is this clear? Okay. How to do this thing in JASP? So this is JASP. It has a similar interface. It's no coincidence because the JAMOVI and the JASP people used to be the same people, the same team. And then they had a disagreement on how much Bayesian it should have in it. So they split and they took a different path, which is generally good. There's a little bit of a competition between them. But actually, there's like a bunch of things. They have so many things in here that I really recommend that you go and check it out. JAMOVI is more of a community effort. So many of the modules are not done by JAMOVI. In JASP, most of those are done by the JASP team in University of Amsterdam. I'm going to open exactly the same thing. So if we go here to the data library and look at ANOVA, I'm going to open once again tooth growth. They already, as an example, included like a description and telling us what each variable is and even references. And they already ran like the analysis and added a bunch of things that make it very, very nice. We can add, in addition to this, they have all sorts of, I don't know if you like rain cloud, but I think it's just amazing that they did this. Yeah, so if you like rain cloud, which like does this very, very nicely for you, to be able to compare all. Yeah, marginal means you can have a bunch of tables. So it's the same interface. But what you also have here is classical and Bayesian, and if you do the ANOVA, same kind of idea, but it just gives you a little bit more flexibility. It gives you more things that you can do in the post hoc. But whether you use JAMOVI or JASP, you'll see that it's basically the same thing. Here as well, you can shift between if you want to quantify the alternative or you want to quantify the null, and then how likely everything is in regards to that. So very, very simple to do that. Both of them also have equivalence testing. So equivalence testing is the null hypothesis significance testing way to look at whether there are null findings or not. The way that equivalence testing works is that we want to say, instead of trying to reject the null hypothesis, we're going to try and reject differences from an effect size. So if, for example, I ask you, what is a meaningful effect for you? And you say coherence D of 0.2, that's a meaningful effect for me. If it's less than 0.2, I don't think that's an effect. So for me, this is just the null hypothesis. So what you can do is test the effect instead of against 0, you can test it against 0.2. And then if it's less than 0.2, you can say, okay, for me, it's as likely as the null hypothesis. Okay, so it's a trick. Instead of just comparing against 0, it's comparing against some kind of an effect. So this is what it looks like. But here you'll see that there is something called the toaster, and here you can do an independent samples t-test. So basically, we'll include them again. And then you need to say against which effects do you compare this to. So for them, the default is 0.5. They're saying if it's less than 0.5, I don't want to know about this. I think this is a little bit too like lenient. So you can just like change it to 0.2. And then you can see if this is the comparison against 0. So this is what it shows as against 0. But it also tests the null hypothesis significance testing against an effect of 0.2 and minus 0.2, and then gives you the T for that. Anything else I can do here? Yeah, also gives you like a bunch of plots. Yeah, so just basically keep it simple. Replications first, extensions later. Make sure that you don't add too much, and when you add something that it has a really good reason for it. Good. If you have any questions, write me on Slack. If not, have a good reading week.